Okay, so in the last two, excuse me, in the last two videos, we've looked at the mechanisms by which urine is developed along the pathway of the nephron tubule. Our job in this video is to understand how it is, or what is happening along the way, how those mechanisms are interacting to produce the concentration and the volume of urine that we make. So here we get started. So when we're going to start a little bit out of order, just to try to make some sense of this, but where we're going to begin is looking at what the urine looks like once it is in the distal convoluted tubule. Okay. Once the tubular fluid makes it to the distal convoluted tubule, the fluid in it, the developing urine, is hypotonic. That means it has a greater water concentration than all of the fluid in the interstitial spaces around that tube. So we have a hypotonic, more dilute urine than interstitial fluid is. And it's because of all the work that's happened in the nephron loop to remove the ions that are found in the urine. So if the tubular fluid is hypotonic, then that means the interstitial fluid that surrounds the tube is hypertonic. The distal convoluted tubule is, under normal circumstances, mostly impermeable to water. So if there is no other action taken on this urine, the resulting urine that we would make is hypotonic. Okay, so how did we get this dilute or hypotonic fluid there at the distal convoluted tubule? And it's the result of countercurrent exchange, also known as a countercurrent multiplier. So it's countercurrent exchange that occurs between the descending limb and the ascending limb of the nephron loop. So if you imagine the nephron loop, if you imagine the nephron loop, and I have lost the capacity to be able to draw on the slides, so for that I'm sorry. But if you can picture the nephron loop, there's the descending limb that comes down, and then the ascending limb that goes back upward. So the fluid moves in opposite directions in the two limbs. In one side it moves downward, on the other side it moves upward. That's the countercurrent piece of this countercurrent exchange. And because of this process, we can see passive water reabsorption from the descending limb that is caused by the osmotic gradient that we produce in the kidney because we're pulling salt by active transport from the ascending limb. So it's going to require for you to really get this idea of countercurrent exchange. And this is an important concept. It's important because it is so efficient at removing water and at moving salt along the concentration gradients. It's so efficient that we see countercurrent exchanges frequently throughout the body. Especially when, if we did a comparative study, if we were looking at other animals to see where countercurrent exchanges occur, we see it in places where we conserve heat, in places where we conserve water. They're very, very important processes, these countercurrent exchanges, or sometimes referred to as countercurrent multipliers. Okay, so how does the countercurrent exchange work? In the descending loop, or descending limb of the nephron loop, this is where we're pulling water by passive means from the tubular fluid. So as we look at the fluid at any point along the descending limb, what we would see is that there is a very small difference from between the concentrations of tubular fluid and the surrounding interstitial fluid. But because the concentration gradient, or the concentration of salts in the kidney tissues, gets more and more concentrated, so we get stronger and stronger solutions, the deeper into the kidney that the nephron loop is found, that region where the fluid is passing through the nephron loop, as we get closer and closer to the center of the kidney, saltiness and other ions, not just salts, but we see that there are greater urea concentrations in the medulla than there are up in the cortex. There's lots and lots of solutes found in the fluid of the interstitial regions. The deeper into the kidney you sample. And so as the fluid goes down the descending limb, these increasing concentration amounts form an osmotic pressure that calls water from the interior of the descending limb into the interstitial fluids, so that we do reabsorption of water as the descending limb makes its way in toward the center of the kidney. And it is such a strong concentration draw, or it's such a, the difference is so marked from inside to outside, that almost instantly, as the fluid is passing through the nephron loop down that descending limb, almost instantly isotonicity is reached at any point along the way between the inside and the outside. But because the concentration is becoming greater and greater, the deeper it goes, we're constantly drawing out water as that fluid descends along the descending limb. So that by the time we get to the bottom of the descending limb, the tubular fluid has become about 4 times more concentrated than it was at the beginning of the descending limb. So that's how significant this concentration gradient is as we get deeper and deeper into the kidney. On the other side, we see again movement of materials from high concentration to low concentration. But this time it's active transport that's pulling out all those salts and other materials that caused the urine to become more concentrated. So that by the time we get back up to the top of the ascending limb, we've pulled out so much salt that the resulting tubular fluid is actually hypotonic as it enters the distal convoluted tubule. Rats. This term should be hypotonic right there. So we do active transport to pull out urea and salts from the fluid in the ascending limb, and the active transport is so vigorous that by the time we get to the top of the ascending limb and enter the distal convoluted tubules, the fluid is hypotonic. Now remember that there are peritubular capillaries that surround the nephron tubule. And in the juxtamedullary nephrons, those that have their glomerulus right at the border between the cortex and the medulla of the kidney, and whose nephron tubules drop deep into the medulla, those guys have a special capillary network that surrounds the nephron loop called the vasa recta. The vasa recta pick up, they have their arterial side at the ascending limb and their venous side at the descending limb. So they are drawing fluids across the nephron loop from ascending limb to descending limb. And they help to maintain the osmotic gradient that we see. As they move across the ascending limb, they grab up all that salt that has been removed from the tubular fluid, and they pick it up, and as they cross over the ascending limb, they draw up all that water by osmosis. So we even further enhance the activity of the descending limb to remove water because we're passing very concentrated blood across the ascending limb. It draws the water that's come out of the descending limb into the capillaries and then away from the kidney, so that we are constantly depleting the water in the interstitial fluids around the descending limb, so that we can pick up even more water from the next glop of tubular fluid that is passing down the descending limb. So the vasa recta do this work of being the sink, if you can think of it that way. You think of the source of water is the descending limb. The sink for water, the place that water is leaving the kidney, is through the vasa recta. And so the saltiness of the vasa recta draws water from the interstitial fluid, which then reconcentrates the interstitial fluid and draws water from the descending limb. So draw pictures. One thing that I recommend is that you draw pictures showing lowest concentration in the vasa recta of water, so that it becomes the place that the water goes to as it's drawn by osmosis out of the urine. Okay, so how do we control urine concentration and volume? One way that we control is through hormones, and we've talked a little bit about antidiuretic hormone. It is secreted by the posterior pituitary. The signal for the posterior pituitary to secrete is when the blood becomes concentrated. So if there is low water concentration in blood, or if the blood volume overall becomes low, antidiuretic hormone gets secreted by the posterior pituitary. In response to ADH in the blood, the distal convoluted tubule becomes permeable to water. Under normal circumstances, water can't move across the distal convoluted tubule across the walls of the DCT. But with ADH stimulation, water channels open up because we insert those water channels. The water channels are called aquaporins. I introduced that term to you last semester. But these are just little channels, little passageways that water can flow freely through. And so with antidiuretic hormone being produced, then the distal convoluted tubule puts aquaporins into the tubule wall, and water then is reabsorbed from the tissue fluid into the from the developing urine into the tissue fluid. And yes, from the distal convoluted tubule into the tissue fluids. And so as a result of antidiuretic hormone, we get a low volume, very low water, so therefore very concentrated urine produced. So antidiuretic hormone opposes diuresis and creates concentrated urine because it allows us to draw fluids out of the distal convoluted tubule and even across the collecting duct some. Urea and uric acid, we talked about them briefly. Here's where I get to tell you about them specifically. Urea is the product of metabolizing amino acids for energy. So when we break down an amino acid, remember there's a nitrogen amino group on one end of an amino acid. It's got nitrogen in it. Nitrogen, when it's metabolized, produces very toxic nitrogenous compounds. Ammonia is one example of a toxic nitrogenous compounds. And we do our best to make that non-toxic by converting ammonia into urea, but it is still, even urea can be toxic. It's an important solute in the kidney medulla because rather than peeing away all of the urea, we actually place urea in the tissue, in the kidney tissues, and it acts as a means of creating osmotic pressure in the kidneys that draws water out of tubular fluid. Uric acid is the metabolic byproduct of using nucleic acids as an energy source. And there are two nucleic acids that when we process them, we result in having lots of uric acid left over. Those two are called adenine and guanine. So those are two different nucleic acids that if we process them to get energy from them, we'll get uric acid as a byproduct. Interestingly, as the uric acid is passing through the proximal convoluted tubule, we reabsorb 100% of it. Comes back into the tubular fluid, and later on in the distal convoluted tubule, we secrete about 10% of the uric acid that we reabsorbed out of the filtrate. So we filter 100%, we reabsorb into the body 100%, and then we secrete out about 10% of the uric acid that we produce. And the rest is found in the kidney, where it maintains a concentration gradient. So it also, like urea, helps to maintain that concentration gradient, osmotic pressure of the kidney. So by the time we have produced urine, here's what urine looks like. It's mostly water, about 95% water, and the rest is waste materials. Urea and uric acid are important components of that waste material. Creatinine, as we break down creatine, one of the next products that we get while we're breaking down creatine is material called creatinine. Remember, creatine phosphate is important in muscle function. Creatinine is also a waste material. There are also going to be trace amounts of electrolytes. We'll have reabsorbed a lot of that, but there's a little bit left that we can't deal with, or that we may be trying to maintain homeostasis in body fluids. So we've got to excrete those electrolytes. And then sometimes amino acids slip through and are found in the urine. So that average urine production is somewhere between 1/2 liter to 2.5 liters of urine or pee a day. It's about 1/2 gallon. We're able to produce something on the order of 50 to 60 milliliters of urine per hour. If we're producing less than 30 milliliters, then that can be an indication of kidney failure. The renal clearance time, remember there's a renal plasma threshold. That's the amount that we can remove from urine of a material as it passes through the nephron tubule. Renal clearance is the time required to remove a particular chemical from the plasma to see the complete reabsorption or tubular secretion. This renal clearance is the time required to get a material out of blood plasma and to get it into urine. And we use renal clearance as a means of testing the capacities of the kidney to function. So we can give somebody a little dose of inulin, which is a complex polysaccharide like a glycogen, but rather than being something that is digestible, it's indigestible. We filter it. We do not reabsorb it or secrete it after it's been filtered from the glomerulus. And so we can get a sense of, or calculate exactly what the glomerular filtration rate is by taking blood samples over time. So you give somebody some inulin, a half an hour later, you check their inulin concentrations in their blood. That's probably about when it would be the highest. And then you can watch it decrease in amount of that inulin over time until there is none. And that would then tell you the glomerular filtration rate. Creatinine can be used the same way. It's also filtered but not reabsorbed or secreted. And so it can also be used to calculate glomerular filtration rate. That's what this term here is, GFR, glomerular filtration rate. And then last of all, para-aminohippuric acid, PAH, plasma clearance test can be used to determine the rate of plasma flow into and then out of tubular fluid. So it's got a little bit more specialized means of telling how kidney function is going.